All right, so we're going to hear about a scrappy time series based out of Newport, Oregon, the Newport Hydrographic Line. So Newport, Oregon is a home port for many commercial fisheries, We all know that Dungeness crab is the highest value fishery out of Oregon. It's 51 million per year. But what is number 2? Go for it. Nope.
Salmon.
Nope.
Clam.
Nope.
Tuna.
Nope. No.
Sardine.
They're little. Not sardines.
Okay, they're not boating.
Mink shrimp. All right. We got a can of tuna and a bottle of wine.
You can advance your slides using this, or the keyboard, and here's the
And
right laser
thank you. Our
pointer.
next speaker is indeed Jennifer Fisher, who comes to us from the Northwest Fishery Science Center, based out of Newport, Oregon, and she'll be talking to us about the some of the important findings that we get from the Newport hydrographical line and the time series that's been going on there for about 20 years.
That is not a scrappy time series, I would like to clarify. Yeah, so good morning, everybody. As you said, my name is Jennifer Fisher, and I work for the Cooperative Institute of Marine Resources Studies at Oregon State University. And this is my first time at OCB. I'd like to thank the organizers and the organizers of this session for inviting me. I'm really excited to tell you some stories about the West Coast. Heidi set the stage perfectly for my talk. You'll see a lot of common threads in what she was discussing and what I'll be pointing out here. I'd like to start by acknowledging my co-authors, especially Bill Peterson, the late Bill Peterson. He sadly passed away to cancer last fall. He's really the brainchild of this time series. He was a NOAA senior scientist, and he started this time series in 1996, and it was really through his continued efforts to keep this alive that I'm standing here today to show you these data. I'd also like to acknowledge Kim Jacobson, who's also from NOAA, and Shuning Du and Sam Zeman, who are also in our research program now. They're both from Oregon State. And then the previous folks who have really put a lot of time and effort into collecting these data. It takes a village. So as Heidi mentioned, we're also interested in basically how the physical properties are driving food web structure. And we're looking at the physical properties in different scales. So the scales that are important to us are basin scale properties such as the Pacific Decadal Oscillation and El Niños, and then also the local physical forcings such as the seasonal cycle of upwelling and downwelling, how that affects the coastal currents, and the timing of when that upwelling happens every year. And we're really looking at this from a bottom-up process. We're looking at the phytoplankton and the zooplankton, and we're trying to infer how that's then, energy is transferred up the food chain so that we can forecast fisheries. And ideally, we would understand the mechanisms that are driving these arrows. Right now, we're really more looking at correlations, and we're trying to work more closely, or working harder to understand the actual mechanisms of how energy is being transferred up this food chain. Today, I'm mainly going to focus on copepods and krill, so it was great that Heidi was focusing more on phytoplankton. And again, we use a suite of both physical and biological indicators to try to basically forecast fisheries. And so that's juvenile salmon recruitment of sardines, sablefish, and rockfish, and then other predators such as adult sablefish and adult salmon. So the data that I'll be presenting today are from the Newport Hydrographic Line. This is a time series that started in 1996. It's a very high-frequency time series. We go out and we sample 7 stations twice a month. Those stations are located on the shelf and slope from 1 to 25 miles from shore. We do this year-round, and so even though it is a single transect, it's a very high-frequency sampling. At each station, we collect standard hydrographic measurements, CTD. We don't have a rosette on our small vessels, so we only collect surface chlorophyll and nutrients. And then we tow a variety of different nets to target phytoplankton, zooplankton, larger zooplankton such as krill, fish larvae, invertebrate larvae, and pteropods. When ship time and funding allows, we also try to put this time series off of Newport into a broader geographic scale. And since 1996, we've been sporadically sampling these transects along the West Coast, spanning from northern Washington down to California. And just for reference, here's Newport there, where we have the high-frequency sampling. And again, this is really largely fluctuated based on funding cycles. So during the Globeck years, from 1997 to 2003, we mainly occupied these transects from Newport down to just north of San Francisco. From 2003 to 2011, we were allocated NOAA ship time, and we tried to sample the entire coast, but again, that wasn't as frequent. And more recently, we focused back on these transects that had the higher frequency from the Globex years. So all in all, we're starting to amass a time series of a broader geographic region, and we have 12 years from the spring, 8 from the summer, 10 from the fall, and 4 from the winter. So we can use this information to put our high-frequency time series into a broader geographic scope. And when I put this talk together, we talked to the organizers, and we were talking about how it's really difficult to take these time series, and especially the biological observations, and relate them to management. And so I wanted to just kind of highlight some of the management drivers that we use to try to address some of the questions, some of the research priorities of especially NOAA, and how we put our data into this context. So bear with me while I walk through just a few of these management drivers. The main one that we address is ecosystem-based management. And really that's a lot of what we do, and that's basically describing species distributions, abundances, their fluctuations over time, how that relates to the physical environment. Ideally, if we have a good enough idea of how the physics and the biology are coupled, we can develop habitat suitability models. And then if we understand some of the thresholds, say temperature thresholds or OA thresholds, we can then develop vulnerability and risk maps, and over time assess resilience of the ecosystem. And as Heidi mentioned, this is a very complicated and difficult task, but it's what we're really moving towards. And then another management driver is ecosystem-based fisheries management, and that's basically putting everything in the previous bullet into action. So how are those species distributions changing, and how can we then make management decisions based on those? And I'll show you some examples of some ocean indicators that we're using, and we're also conducting some sibling regression models for ocean salmon survival that I will not be talking about. Another management driver is basically sustainable and safe seafood. So this is where stock assessments come in. So how can we maintain sustainable food for the future and not have fishing declines? I won't be talking about this, but this is also where understanding and being able to ideally forecast harmful algal blooms comes into this management driver. And lastly, how is all of this going to change in the future? How are these species distributions and interactions going to change with increasing climate variability and climate change? And of course, ocean acidification and hypoxia and this idea of multiple stressors is a really important point here. So by the end of this talk, I hope that you come away with how we're kind of addressing each of these through a few examples from our data. Ooh, that did not work out. That's going backwards. Uh-oh. So there's supposed to be a temperature plot under here. So as you can see, there's a lot of temperature, there's a lot of variability in the temperature. That's too bad. So this was supposed to be a monthly time series analysis, or a time series from 1996 to the present, showing basically temperature and salinity fluctuations. And the take-home message here is that basin scale forcing, such as positive PDO, downscales to the local event scale. So during positive PDO, we actually see warm and fresh water on the shelf. During negative PDO, we actually see cool and salty water on the shelf. And then I wanted to also point out there were 2 periods where we had different water types on the shelf, but you're not going to be able to see that here. So one of the main questions that we try to look at, again, is how do basin scale, regional, and local physical drivers affect primary and secondary production? And I'm only going to be talking about secondary production today. And as we all know, the strongest cycle in production is seasonal. And so this plot could be phytoplankton, it could be temperature, it could be nutrients. But here I'm just plotting the climatology, and the orange bars are the number of copepod species on the shelf, so the species richness. And you see a strong seasonal cycle where in the wintertime we actually get a lot more species on the shelf, and it's during the summer upwelling months where we actually have fewer species. And this pattern is driven by the seasonal cycle of upwelling and downwelling. And this line represents the actual copepod biomass. So even though we have fewer species during the summer, those species are larger, and so we actually have a higher copepod biomass during the summer on the Oregon shelf. Oh no. This is terrible. It doesn't show on my screen either. I do have my laptop. Let's plug you in there.
Oh,
Do you need an adapter?
USB to, do you have an adapter?
Let's take here too. Take a moment, drink water. I'll get you some. I paused this one too. Thank you. Let me just close everything so you don't get any pop-ups.
Actually,
where do you wanna
I'm gonna
start?
go back. Yeah, let's start here. I'll just briefly go through it. Okay.
It's weird. Thank you. Got it. Okay, go for it.
Oh, I know.
Thank you, everybody. So I'm going to briefly go through this. So here we have the PDO and the ONI in the top panel. And the middle panel is a temperature anomaly from 1997 to the present. And you can see, as I was mentioning, these really strong positive temperature anomalies, especially during the 97-98 El Nino, during the 09 to 10 El Nino. And I really want to point out the strong temperature anomalies that we observed during the last marine heat wave or anomalous event, dubbed the warm blob. And here's the beauty of time series. We can see now, then put this recent anomalous event in context to the last 22 years. And we actually see that those temperature anomalies exceeded even the really strong 97-98 El Nino. So again, during positive PDO years, we basically get positive temperature anomalies and fresher water on the shelf. During negative PDO or La Nina years, we get negative temperature anomalies and saltier water on the shelf. And then here, there's a mixed signal where the PDO is oscillating from positive to negative, and we have generally a positive temperature anomaly and salty water on the shelf compared to this period where we actually had a strongly persistent negative PDO, and we get negative temperature anomalies and a mixed salinity signal. So we talked about the seasonal cycle, and if we take those species and we actually look at the copepod community structure, here I've put all of the copepod species for every sampling month into a multidimensional scaling plot. And each dot basically represents the copepod community at that sampling period. And there's a lot of scatter and there's a lot of variability. And if you squint, you can see that, over here we have a lot of pink and blue triangles associated with 2016 and 17. So the community is kind of clustering together there. But you can't pick out too much of a pattern. Interestingly enough, though, the x-axis actually represents 76% of the variability in this plot. And so if we take those scores and we plot them over time, we actually see that, again, the community composition of these copepods on the Oregon shelf are oscillating on this seasonal cycle. And so they're oscillating from a warm community to a cold community. And that's essentially happening during the upwelling, downwelling cycle. And so you see in the winter, we get this warm community. In the summer, we get this cold community. A few exceptions to note are during the 97-98 El Nino, we didn't see that shift to a cold water community. And similarly, during 2015 and 16, we did not see that shift to a cold water community. And I'll explain the implications to the food chain later. So similar to what Heidi did, if we remove the seasonal cycle from this plot, now I'm plotting the monthly anomalies. And now we see that the community is oscillating on a much longer time scale. And so we're getting positive anomalies or warm communities, and then oscillating to cold communities. And if we plot this next to the PDO, we see a strong concordance or a strong correlation between the two. And if you run a cross-correlation analysis, the correlation is about 0.65. But it's important to note that the biological response to the physical forcing of the PDO lags at different time scales. So we can't just use the PDO if we want to understand the biological response, because the lag is based on basically the intensity of the event. And I'll explain more of that later. Here, I've just replaced this top plot, which was the copepod community composition with actual species richness. And I just wanted to point out that it's during these warm periods or positive PDO that, again, we get increased copepod species off the Oregon shelf. And here, the anomalous event from 2014 to the present actually stands out amazingly here in this plot. And these copepods are drifters, they're plankton, and so we can basically use them, and they have known water mass affinities, so we can kind of use them as biotracers. And we actually got an increase, an almost double fold of copepod species to Oregon during this recent event. And so this indicated that that water mass that reached the Oregon shelf basically came from a region that we had not observed before. So we can use these guys as kind of biotracers. So while I didn't show it, there's actually no relationship with upwelling in the copepod community structure, and so it's really these basin scale processes that are driving the fluctuations in the community. And again, there's variable time lags between the biology and the physics, so it's very difficult to model. So another question that we're very interested in is how do these fluctuations in secondary production affect higher trophic levels? And at the beginning of this talk, I was talking about trying to get at the mechanism, and one of the mechanisms there, so we know, for example, that interannual differences in copepod species composition do correlate to, say, salmon returns, and one of the mechanisms might be actually the bioenergetics of those copepods. And we know that the warm water, or what we call the southern species, are actually smaller, and they have very little lipid stores compared to the cold water species or boreal species. And they have really high wax esters. They use them as an overwintering strategy in boreal climates. And here's just two of those northern copepods, and you can actually see the lipid sac in this animal. So basically, when we have these large climatic forcings, we're actually altering the bioenergetics of the food chain. And just to drive that home a little bit, here's, we collected plankton samples and we ran the fatty acid composition and the lipids classes of the entire plankton sample. And here, green is plotted, or summer is plotted as green and winter is plotted as black. And you can see that the fatty acid composition between the summer and the winter community is different. And that's mainly driven by diatoms in the summer and a more dinoflagellate-dominated community in the winter. And if we then plot the recent warm events, the blue triangles here are actually the summer of 2014, before that warm blob water came onto the Oregon shelf. And you see that those fall within a normal kind of summer fatty acid community. But then these red dots are after the blob water intruded the shelf, and we actually see that that water brought a zooplankton community that was even further outside a winter community. And I'm not going to show the lipids, but this community was lower in lipids compared to other winters. So how will the trophic levels be affected in the future? We know that large-scale climatic forcing is becoming more variable, and we also know from our 22 years of data that the intensity of that warm event or that perturbation is related to the time it takes for that ecosystem to quote-unquote recover. And so here I'm plotting from 7 El Nino events, the El Nino intensity. So that's not only the SST anomaly at the equator, but that's also multiplied by the number of months that that anomaly existed. And I'm plotting that versus the time that we observed these lipid poor or warm water copepods. And we see this strongly linear relationship. And it's important to note that the 09 and 02, 03 El Nino events were actually very short in duration, but they were very intense. And so these biological systems are responding not only to the temperature anomaly, but basically to the length of the system. So you can have a very intense or a very strong temperature anomaly, and you might see the same perturbation or the response in the biology as you would see from a prolonged lower temperature event. And if we put the recent warming on this plot, here's just looking at the SST intensity from 2014 to 17, and you can see we're way outside the box here. So it's pretty scary. What we don't know is how is the resilience of this ecosystem, and how will it fare under increased stress and variability. And I like to think of it as kind of like the memory or the hysteresis of the ecosystem. At what point will these communities not be able to recover and come back to normal? So moving into a larger zooplankton, we're also interested in what are the environmental drivers of krill abundance and distribution. We probably all know that the krill are important prey for large predators such as whales and marine seabirds, but they're also important prey for commercially important species such as juvenile sablefish in the Northern California current. And on the left here, I'm just plotting, these are from our broad scale surveys, the abundance and distribution of Euphausia pacifica. It's our most common krill off the coast. And the size of the circle depicts the abundance of that at that station. And it's kind of difficult to pick out, but basically we have the highest abundance along the shelf break here. The bathymetric line there is the 200-meter isobath. And here on the right, I'm plotting the abundance and distribution of another species. This is the coastal krill, Thysanowessus spinifera. And we see that they're not distributed over as large of a geographic range, and they're also found closer to the coast. And if we actually look at kind of a probability prediction map, we can see that Euphausia pacifica is basically, again, along the shelf break and at canyon heads, and Thysanowessus spinifera is at the inner shelf. We've just gotten some funding for a graduate student actually to look at more of these data. These were only from up until 2006, and so hopefully they can quantify more of these samples and flesh out this data. So ideally, we will be able to develop habitat suitability models for these krill, and then ultimately be able to use that to forecast predator distributions and or reduce whale entanglements, which have been increasing in numbers over the last years. So how does krill distribution affect higher trophic levels? These two species vary quite considerably in their lipid content. And so here I'm actually plotting the wet weight versus the total lipid of the animal. And you see that the red dots here of Euphausia pacifica, they're all kind of hovered, clustered around about the same. And then the T-spin adults, they have about the same lipid content as Euphausia pacifica, but it's actually these T-spin juveniles that are really packing on the lipids. And so if we really want to understand energy transfer up the food chain, we need to better understand the distribution of this actual life stage, not just the different species. And it's important to note that this lipid-rich species, Thysanowessus spinifera, was basically absent from our samples over the last warming phase. And so it's important to think about how will future warming events, how will future climate scenarios affect these critically important prey. So how can ocean ecosystem indicators forecast fisheries? That's the goal of, I think, a lot of monitoring programs. And we've developed this method where instead of looking at single indicators, we try to basically look at a suite of indicators. And here we've tailored them for salmon returns. And so the indicators are here. We have a few basin scale indicators, the PDO and the Ocean Nino Index. We have some regional physical indicators such as the upper mixed layer temperature, sea surface temperature, and salinity. And then a suite of biological indices. These are our copepod metrics, the community composition and abundance of fish larvae, as well as juvenile salmon catches from a separate survey. And what we've done is we've averaged these indicators over the critical life period of when it's important for these juvenile salmon. And the idea here is that when these fish first go to sea, it's really important that they have food and good ocean conditions for their survival. And it's at that point when it's setting their survival. And so we've averaged these indicators at that point when those fish enter the ocean. And we've then ranked them and color-coded them based on whether it's poor for salmon, which is red, or positive for salmon, which is green. And it's a very simplistic approach, but it's something that managers can easily look at, and they can say, okay, 1998 was a poor year for salmon. 2003, 4, and 5 were pretty poor, and 2015, 16 were quite poor. And so it's very simple, but it's very effective visually. And then we take those indicators and we run a PCA. And again, we've looked at individual metrics such as copepods or the PDO or temperature, but it's really when we capture this suite of indicators, we're able to explain a lot more variability in fish returns. And so we take the first principal component of those indicators, and here it's plotted against the actual adult salmon returns of Chinook salmon, spring and fall Chinook salmon, and then coho survival on the bottom. And you see that there's a pretty strong relationship here. And so now we can use that information to forecast fisheries, especially for salmon, since these fish for Chinook, they spend 2 years in the ocean and coho spend 1 year. So the juveniles come out, we can say ocean conditions were good or poor, so we would expect high or low returns 2 years later. And these are fish that are returning to dams in the Northern California current. And I just want to point out that we are looking at indicators from a single station, but we're really indexing ocean conditions on a broader scale. And so here I've plotted that same principal component of the indicators that we actually tailored for the salmon. And just as a proof of concept, we did this for sablefish in the Northern California current, rockfish from the Central California current, and sardine recruitment from the Southern California current. And this is in its initial phases, and we'd like to develop this further where we could actually tailor these indicators to each of these stocks. But it's just to show that we're really capturing a lot of the variability in a much broader scale. And one of the biggest challenges that we've had is how can we then take this information of ocean conditions and incorporate it into stock assessment? And I think that's really where we need to go. So moving on to the climate science, I couldn't really not talk about hypoxia and OA. So through our monitoring, of course, we're interested in whether waters are decreasing in dissolved oxygen. Here's a modeled plot where, as we all know, we're expected to get large declines in dissolved oxygen. And this is actually from a GFDL model from 200 meters off of Newport. And these are our data from a similar location from 1999 to almost the present. And we see oscillating trends. So we see a negative trend until about 2005, then a positive trend until about 2009, and then things have been pretty stable since then. But again, we're right at the beginning of this negative curve here, so it's going to be really important to continue monitoring the oxygen in the future to see whether we're actually observing what the models are saying. Similarly, with ocean acidification, we're in the middle of the decline of the pH curve, as according to the model data. And we know that already on the shelf off of Oregon, our waters that upwell onto the shelf are seasonally corrosive. So here I'm just plotting aragonite saturation, the climatology of aragonite saturation. And we see that during the upwelling months of June through August, we have water upwelled onto the shelf that is at an omega 1 or below. So this is corrosive water already being upwelled onto our shelf. And if we look at the depth time profile of aragonite saturation off of the slope, I've just coated the omega-1 in black here. And you can see over time that horizon has been shallowing, then leveled off, becoming deeper, and then shallower again. So again, in these changing climates, it's going to be important to continue monitoring this to see whether ocean acidification is impacting our ecosystem in the future. And right now, we're working towards looking at these aragonite saturation values and correlating that with limacina to see if we see an impact. So I just wanted to finish with a few challenges that we've encountered. I hope I've given you just a flavor of how we're trying to address some of the research priorities of NOAA. But again, we don't want to just address the research priorities, we want to actually put this information into action. And so we're really trying to move from correlation into mechanisms so that we can understand the energy transfer of the food chain. We think that that will be very important. It might be transferable to other locations. And ideally, we would incorporate our ocean monitoring data into stock assessments. Funding has been a huge challenge. We are not NOAA line item funded, and we are barely funded. NOAA supports us right now, supports 1.5 people and our ship time. And so we are constantly writing proposals and trying to interact, leverage partnerships with other people to continue our data set. And then finally, this might seem like a strange bullet point to put on a final slide, but if you're going to maintain a time series, you're going to have personnel changes. And if we're actually counting the biology, we're not just collecting temperature and salinity. It's important to make sure that those people are trained and that there's a lot of overlap, so that if you see a shift in, say, species richness, that's actually because you're seeing a shift from the environment and not just a shift in personnel. So I can't impress that enough. So that's basically it. I'd like to thank our funding sources over the years. We put all of our data on the Northwest Fisheries Science Center website. We also put our salmon forecasts up there. And we maintain a blog where we also post data and talk about recent ocean conditions.
One quick question, and then we'll take a break.
So I really enjoyed your talk as somebody who has a tremendous interest from the satellite world in higher trophic level retrievals. My question is, your ocean ecosystem indicators that you're using for all of your analyses, are those based on data you have or data you want?
They're based on data we have.
Okay, so your list of what you would want would be different perhaps, or?
It would be, and it would probably be different for whatever species we're looking at.
Do you have that written down anywhere?
Of what we would want?
Yes.
I can email you what we would want.
I've been in the government long enough where I have seen more people do and redo plans. Here are the essential ocean variables. Let's write those another way. Let's list those another way. We can process and parse information in biological and biogeochemical and ecosystem variables any number of ways, and we tend to repeat ourselves after time, right? Mm-hmm. So I'm interested in finding some consistency in things like ocean ecosystem indicators and what people are actually looking for, not just in situ, but so I can do the remote piece too. So that would be really helpful.
Yeah, that's important, and I'll just make one quick comment. I think that it's important to know too that depending on the species that you're looking at, those indicators may change. And so it's going to be very difficult to just find one suite that will be applicable to all systems and all species. So.